Good afternoon, everyone. Thank you for attending our webinar this afternoon. My name is Karen McLeod. I'm the manager of MP Biomedicals Australia, and it's my pleasure to welcome you to our session this afternoon, where our research scientist, Dr. Yongxing Tan, will present on some of the new options that we're offering for overcoming DNA extraction challenges in microbiome research. Dr. Tan is based in our Singapore facility in MP Biomedicals under the Life Sciences Molecular Biology Research and Development Department. And he's going to highlight how to overcome the various challenges faced in DNA extraction using our latest SpinEasy and MagBeads technologies. He will share his passion and knowledge of these new technologies, which you can use to solve the key challenges in your microbiome research. So just before we start, I'll just let you know we'll be muting all microphones for the duration of the talk, but you will be able to post some questions. So that you should see the Q&A panel on the right of your screen, and you can ask any questions throughout the presentation by clicking on the purple button at the bottom of the panel. So please send your questions throughout the presentation, and then Dr. Tan will be able to answer most of them for you at the end, and anything that we can't answer, we'll certainly get back to you in a short while. And finally, we'll be offering free sample kits to thank you for your time today. So stay on until the end of the presentation for the details of how to get your samples. So without any further delay, we'd like to welcome Dr. Tan. So we'll cross now to Dr. Tan for the presentation.
Hi, thank you, Karen. Thank you, everyone. My name is Yongxing. I'm based in Singapore, MP Biomedicals. So I will be a presenter for today's webinar. I'll go through some of the agenda that we have for today. First, we have an introduction to the microbiome research, and we'll follow by challenges in the DNA extraction. And then we have some solutions for you. We have some methodologies from MP Biomedicals, which consists of two kits that we have recently launched. This is the SpinEasy DNA kit for soy samples, and also SpinEasy DNA kits for feces. Another one is the MagBeads Fast DNA kit for feces. And at the end, I will summarize the microbiome research workflow and also our products, and followed by Q&A. So I'll now go into the introduction of microbiome research. So first, we want to understand the significance of carrying out such microbiome studies, microbiome research. First is to understand the structure of the environment. We want to understand how to characterize the species diversity, as we know that microbiome research focuses on the behavior, interactions, and functions of the microbial communities within a specific environment. So this is important to understand different landscape, different types of soil. So microbiome research can assist in achieving such outcomes. Next is the comparative studies. We also want to use the microbial communities under different factors. For example, a healthy subject comparing to sick subjects. So they can come across as biomarkers, and microbial communities can serve this function. Thirdly, we can use it to identify new species, new genes, and this is a very interesting area, as the recent studies have shown that there are tiny proteins that have been ignored by the researchers due to the fundamental difficulties in detecting them. But now, due to advancement in science and also microbiome research, we can now understand these novel proteins. We can help to facilitate drug development, which is an area of biology for study. So this will give us the new frontier for future therapeutic drug development. Next is the workflow for the microbiome research. First, we want to design the studies. We want to know what kind of samples, how many samples, or how many subjects to have for the study, and the control groups, and what's the outcome that you are targeting. So as a first step to conceptualize a study. Next, then we'll proceed to collect the samples. During this process, we need to understand how to collect them, understand the biosafety and handling of such samples. Transportation, storage of samples are also very important, as this is the steps that can preserve the integrity of the samples that you are dealing with. And then next, which is our focus for today's webinar, which is our DNA extraction. This is where we understand the process of cell lysis and also a couple of procedures to obtain your pure DNA for analysis. And analysis will come from the next step, DNA sequencing. So we will use PCR amplifications, metagenomics sequencing. So these are just a few techniques to understand your DNA and also analyze them to achieve your outcomes. And next, we will, after the sequencing, we'll have the read classifications. So once you obtain all this sequencing, we will want to have the identification of the reads of the microbes. And then we proceed on to have a statistical analysis to be able to tell what's the significant difference, or is there any slight difference between your samples or in the environment. So next is the DNA extraction. So this is the overview of how you have your samples and also obtaining your pure DNA. So first you will have samples from animals, mice, bovine, and also plants. So these are the samples that you can retrieve, and then you put them through a cell lysis process where you can unlock all the intracellular component, ideally the more DNA and also the RNA for analysis. And you go through a series of purification, and that will be your pure DNA for analysis. Going into the principle of DNA extraction, you have a lysis step where you unlock all these, release all these intracellular components, the proteins, RNA, DNAs, and at the same time, you also release some of the other contaminants, which leads to the next step, that you purify them, you remove the debris, you remove the large particles, precipitate the protein, and if you are targeting DNA extraction, we want to remove the RNA as well. And then the next step is to select a suitable matrix to bind the DNA, and lastly, is to wash off all these unbounded substances, and what is remaining is your pure DNA, and you will elute them in pure form. Now we will look at some of the applications that you can use for microbiome research. So microbiome research is, there's many areas that you can use it in. First is the agricultural application. This is where you can use it to characterize soil microbial communities. And due to its stability, DNA is often the choice to characterize microbial communities in soils. So successfully, people has been using it to analyze the soil, and also it depends on the quality of the DNA extracted to reflect on the soil fertility. And also people use it to characterize the fungi, the bacteria that's present in the soil, because they are the microorganisms that play fundamental roles in nutrient cycling. Next is the healthcare application. People use microbiome research in gut microbiome studies. This is a growing area, and in fact, it's one of the hot research area at the moment. People are getting more information, more useful information in non-invasive manner to understand health effects of humans, and also study of antibiotic resistance, bacterial detections in healthcare. For environmental applications, we have pollution remediation using microbes. This is where you can have microorganisms that, for example, utilize oil as a food source, and you can emulsify them in water to facilitate the removal. Just an example. And for ecology application, we have the monitoring of invasive species, tracking of the seasonal populations. And biotechnology, we have the discovery of new genes, enzymes, and natural products from all the microorganisms. Biofuel applications, there's microfermentation, where living things or microorganisms will have byproducts, and it can be made into biofuel. Next, I'll be sharing on the challenges faced in the DNA extraction. At the moment, in DNA extraction area, there are many challenges that's faced by the end users. So I will just list a couple of them, and this is some of the challenges that I will share today. So first is that the effectiveness of the lysis process. Lysis is supposedly to be able to break the cell wall, release the DNA, RNA, and also the protein, as mentioned. But there are situations where the effectiveness can be low, especially when you deal with tougher samples that have tougher cell wall, and also ineffective lysis of gram-negative bacteria can occur. So in this situation, the effectiveness of lysis will definitely affect the analysis of the sample. So what we want to achieve at the end is the accuracy, a high accuracy of the sample. We want to have good representation, unbiased view of whichever samples that we are dealing with. And next is the temperature requirement. Some of the samples they are heat sensitive, so the control over temperature on the process is very important, so that you do not lose any of the precious DNA or samples that you desire. Next is degradation. This is a commonly occurring challenge. Is that the samples will get degraded because of presence of enzymes such as the DNAs, RNAs. So when there are such enzymes, it will definitely cause the DNA to degrade, and hence affecting the accuracy of the results. Recommendation will be using consumables that are free of DNAs, RNAs to prevent such enzymatic degradation. And next is the contamination challenge. One example will be humic acid. It's a very common component that's present in soil. And these are the components that will bind to the DNA and causing issues for downstream processing for the end user. Next is the low biomass samples that we happen to have at times. So sometimes we do not have samples that are readily having a lot of DNA for us to analyze. So when we have such low biomass samples, it's a challenge for us to explore what are the different kits or in fact the protocols, so that we can still successfully extract sufficient DNA. If not, they will actually hinder the downstream processing. In fact, they might not be able to proceed on further if you do not have sufficient DNA. So, and the next one is the technological limitations. So right now, there are many extraction kits, or even we used to have some conventional way of extracting DNA. There are a lot of limitations, especially on two components that we are focused on. One is the yield and also the purity. So there are certain limitations of the kits that they can give you a certain yield, they can give you a certain purity. So this will also be a challenge for people who are examining DNA, and this will be important for us to troubleshoot, because at the end of the day, yield and purity are very important to us. And last category, it will be the large number of samples. So there are also studies that the researchers, the end users will have to deal with. Your studies require a lot of subjects coming in, then your samples size is big. So this will be a problem to establish the consistency and the accuracy across the large number of samples. And coming to that, also the processing time needed. You can imagine that if you're using a manual process to process the samples one by one, and you have to process them as soon as to preserve the integrity of the DNA once you receive the samples. So processing time is a crucial factor when it comes to dealing with large number of samples. Last but not least is the cost. This is across all the extraction process, particularly in the large number of samples as well, because when you're dealing with large numbers, your costs tend to be higher. So this is a factor to consider on how you want to execute the extraction process, such that your costs are still justifiable, reasonable for your research. So facing so many challenges, we MP Biomedicals, we have thought about the situation, and also we come up with solutions and methodologies so that we can assist our end users in the process. So first, we are talking about lysis methods. Lysis methods can be classified under the non-mechanical or mechanical methods. So first, non-mechanical include the physical, chemical, and biological. So they include different ways of lysing the cells. So we have heat treatment, we have detergent, we have using detergent and also enzymes to break the cells. And now more commonly, people are moving towards the mechanical side, which is the use of homogenizer. And reason being that it's more highly efficient and also it's more widely accepted for microbiome study. So it provides even efficient lysis to tough environmental samples. And to go along with this homogenizer, people are using commercial extraction kits. So these are the different type of brands of extraction kits that people employ together with homogenizer to extract the DNA. So going back a bit on DNA extraction methods. So these are on the left hand side, you can see that there's conventional way of doing it, where you have the phenylchloroform method, where you have two immiscible layers, and using the polarity of the DNA, they will be separated into the aqueous layer and achieving a purification effect. One of the biggest disadvantages is that it involves toxic chemicals, and in fact, it's harder to operate. So now there's improvement made and commercial products are coming out in terms of the extraction kits, and binding metrics are designed to bind the DNA and make the process easier and less toxic. So coming to binding metrics, there are also a couple of them in terms of technology. First is the silica in terms of glass milk suspension. So we have this proprietary silica matrix in aqueous solution used to isolate and purify the DNA. Second type of technology is the silica membrane. We have it in spin column setting, and it can be used to bind the DNA in presence of high concentration of salts. So generally, silica is negatively charged, and also DNA is negatively charged. So we have to establish this cation bridge such that it will actually bind them together and allow the washing step and also holding back your bounded DNA. The last technology is the magnetic beads. We have the iron oxide, which is the basis of the magnetic beads, and also in presence of salt, they will also form the bridge so that you can bind the DNA, and in turn, it can also wash away all the unbounded substances, the contaminants that you do not want, and achieving the pure DNA we want at the end of the day. So a couple of instruments that we have as solutions to go along with the technology. We have the handheld fast prep and also the fast prep, which is a high throughput model. And one of our more commonly used model is the fast prep 24 5G. It's our latest model, come with a touch screen, and it holds a number of adapters. End users enjoy this larger volume and also more stable equipment to assist them in the cell lysis process, and it's effective and about 20 to 40 seconds for any type of samples. So moving into the DNA extraction kit criteria, we understand that not only we want the purest and also the highest yield DNA that we can get, we also want it to be able to handle a wide range of application. For example, for soy, there are many types of soy. So we want it to be applicable to, you can use it on any type of soil and easily obtain the yield for analysis and also the purity. Performance, yield is always a very important aspect. Quantifiable yield, I would say that highest is even more encouraging. And quality of DNA, we want it to be intact. We do not want it to have any RNA contamination causing smearing and affecting the accuracy of your DNA analysis. So on the right-hand side, you can see a diagram. If there's presence of RNA, you can run a gel electrophoresis and see for yourself that is your sample smearing, just as a double layer test to confirm that your DNA is pure before you want to send it for further analysis. Purity in terms of numerical representation. So here we have the use of a spectrophotometer. We use the adsorbance reading of wavelength of 260 and also wavelength of 280 nanometer. So this is a ratio between these two, and the value is supposedly to be higher than 1.8 so that it reflects a pure sample. At the same time, end user will also use the wavelength of 260 and also wavelength of 230 as a ratio to further quantify the purity in terms of a numerical value. So the range should be around higher than 1. In fact, if you can go beyond 2, it will definitely help to reflect that the sample is pure. So at the same time, you do not want to spend a lot of time to extract a sample. So if possible, we try to keep it 35 minutes, or in fact, the faster if you can do it in terms of handling and also your familiarity with your kit, then I think the time can be reduced. And one thing, practically speaking, we also want to keep the cost low for per run, per sample, to make economical sense for your study. Some of the case study or some of the publications done are using one of our kit called the Fast DNA Spin Kit for soy. This is a highlight. I will highlight that the kit has a feature that it can handle the tougher sample, being able to extract high yield. And we have feedback from our end users that they are facing the challenge of low biomass sample, and this is a solution that we provide for them. Using this kit is able to extract the yield, or in fact the DNA that wasn't seen previously, and assist them in the possibility to analyze the sample still. So these are some of publications that result that they use the kit, and these are the results. Sharing a bit more, the Fast DNA kit was cited in more than 7,000 publications across many different types of environmental sample, like the fungi in the glacier context, and then mineral rocks, and many more. So bringing you some of the newer solutions that we have. One of it is called the Spin Easy DNA kit for soy and also for feces samples. So for the Spin Easy DNA kit for soy, we have the commercial product, which is in 50 preparations. We are also happy to supply sample size for users to try out their samples to see the compatibility or to troubleshoot one of the any type of sample that they have in 5 preparation size. As for the feces kit, similarly, we have it in 50 preps and also sample size in 5 preps. Run through a bit of the protocol on the extraction process. So for the soy itself, is a simple 5-step process where you lyse in the presence of the lysing matrix that is being optimized. And we have the lysis buffer, which is to incorporate the conditions to break the cells and also the extract the release of the intracellular component. And then we also apply the RNase A solutions to ensure that we do not want any RNA contaminations doing a DNA study. Purifying it, we will use inhibitor remover S, and this is to remove the proteins and other contaminations through precipitation. And with the precipitates, you can centrifuge it and you can remove it by drawing the supernatant. The next step is to bind the DNA to the spin column, which is the silica column technology that was shared earlier. So this is the step where the DNA will get targeted DNA will get binded, and we are able to wash away the unwanted or all the unbounded components, and giving us the final product of eluted pure DNA. Similarly, for feces kit, they also go through the lysis, removal, binding, washing, elution step. But for feces, we know that there are a lot of unwanted contaminants. We incorporate this special additional pre-wash step that has been proven to be useful, and we are able to assist in removing all the unwanted soluble contaminants even before the cell lysis. So this will help to purify and also ensure that when you're dealing with feces kit, we can give you the high yield and purity desired. So moving into the results. So sharing a bit on the soy samples first. So this was tested across different types of soy. So we have the organic soys, the paddy soys, flour bed soy. You can see in the table 1. And these are the different characteristics that they have. So they have different characteristics. So it is sort of a test for the kit itself, whether does it fit the different types of applications. And in table 2, we have also the purity. So the result shows that the purity are generally above 1.8, and also we have quantifiable yield that you can see numerical values. And also for the 260-230 ratio, we are looking at above 1. And these are healthy figures that we can safely say that the DNA extracted from different types of soy were able to give us the yield and also the purity. And moving a bit further, we want one step further, we will want to check it on the gel electrophoresis. We run the bands, we run the samples on the gel, As you can see in figure 1, our top right corner, we can see intact bands, no smearing, which is a good thing for us. We can see that all this genomic DNA were displayed on the gel, not just in numerical values. And also we want to see whether it can be processed downstream. We use the sample for PCR amplification of a 16S RNA genes across all the different types of soil. And we were able to obtain intact bands again. And so this is a good reflection that the DNA are good for downstream processing. Key slides over here is that we also want to get aware of ourselves on how we stand across to our competitors. So we have two competitors in this graph, which is from the Omega brands and also the Kaigan brand. So we use their kits. We also extracted the same samples and run across a comparison in terms of performance. So we can see that our kit, in terms of spin easy, we are able to obtain similar to the strongest competitor in the market at the moment, which is Kaigan's. But also in some cases, we are actually higher than them, depending on the soil type of sample. So this is encouraging for us because this is our recently launched kit, and we are seeing good performance, in fact, very competitive performance to the competitors. And in terms of Omega comparison, we are already much higher than their yield. And just a point to note is that our fast DNA kit earlier on, I said about the low biomass challenge, and fast DNA kit is actually good for such situation, and it's also proven here that the yield is actually the highest among all the other kits. So in terms of purity, Spin Easy kit for soy is able to obtain generally value higher than 1.8. And this is one of the kits that is able to do that, comparing to the other competitor kits. Of course, noting that the Kaigan kit is able to obtain a figure higher than 1.8, but at the same time, they are also getting figures from higher than 2. From this figure, higher than 2, there's an indication that there could be presence of RNA contamination, because such high values are a value that we actually take note that we do not want the range to cross beyond 2. So ideally, we want it to be above 1.8 and below 2, so that we are pretty clear that this purity is consists of more of the DNA. So there's a chance that higher than 2, the RNA can be present. And looking at the 260-230 purity ratio, we are seeing that Spin Easy kit was able to give a much higher purity, which we are seeing values of 1.6 or even 1.5, higher than the competitor's kit. Moving into the feces kit. So the Spin Easy DNA kit for feces, we are also looking at the yield and purity. So we run it across a different type of species from cow, goat, chicken, etc. So we are also seeing quantifiable yield. The numerical values are there. And then the purity, we are also seeing that the values are higher than 1.8, still below the value of 2. We are seeing values very close to 2, which is very good, because these are the numbers that reflect the purity. And for 260, 230, we are also seeing that some of the figures can go beyond 2. But minimally, we are trying to hit it at least above 1. And again, we test it on the gel. We are seeing genomic DNA bands showing up on the gel, and also intact bands, no smearing, which is good that we do not face any RNA contamination. And again, we run it through the downstream processing, PCR amplification, and all the samples were able to be successfully amplified. And this proved that we can use it across a wide range of fecal samples too. So again, we want to get aware of our kit. How does it compare to the competitors? We run it with the other competitors' kit. This time around, we use another kit called the Tiangen kit. So there are results in terms of numerical yield close. In fact, we are the top. We have the highest numbers in terms of yield, and to ensure that all the yield reflects the running would be ideal. And that's where you have the second figure, which is the gel electrophoresis image. From this, you can clearly see that the band intensity shows up for spin easy kit, which is the B1, M1, and H1, mainly the cow sample, the mouse sample, and also the human sample. But whereas for the other kits, it's a bit of mixed results. Although their yield is showing the figures, but the bands were not that ideal to show up as a bright genomic DNA band to reflect the numbers. So in this case, we are always trying to play safe, that we know that numbers can reflect a set of results, but we also want to check back on the gel to be very sure that what we're getting is the pure DNA. So in terms of purity, we compare across our competitors too. So we have the figures higher than 1.8 and also looking at below 2. So again, we are seeing Kaigan's kit has the tendency to cross beyond the 2 mark, and indicating that there's a presence of the RNA. And for us, we are staying in the range of 1.8 to 1.98. So this is a good amount that we are sticking to. And also 260, 230, we are also looking at a figure from 1 to 2. And in fact, now we are crossing the number of 2. No doubt, Kaigan kit is able to produce some of the higher priority in terms of the mouse sample, but there's always a different type of samples to deal with. So it has to come down to what samples are you dealing with. And also at the end of the day, what purity do you require, and how good is your purity, and also the yield. All these factors will come into play to selecting a kit. So some of the case studies that we have for the Spin Easy DNA kit for soy, we have seen end users from using it in terms of the samples like the river and lake sediments for soy, and also we have seen end users with the plant root soy on the plateau with our Spin Easy DNA kit. Some of the case studies that we have for our Spin Easy DNA kit for feces, not only restricted to just animals, we are also able to see end users using it for feces samples from the insects, such as bee. So this is a very interesting case study. And for mouse, as mentioned, we also test it ourselves. We are able to use this, end users are able to use this extraction kit to test it for intestinal content or study of the gut microbiome. So all these case studies can be found on our website. You feel free to check out if any of the samples will be similar to what kind of samples that you have to deal with. Summarizing some of the highlights for our products is that we proved that the results can be, from the results, we are able to use it across a wide range of applications for different types of soils and different types of stool samples. It has high performance where we see the quality of DNA in terms of numerical yield, and also we also see as intact bands and also no smearing, indicating no RNA contamination. We are also seeing high purity, which is the above 1.8 value, and also trying to keep it below 2 and higher than 1 for 260, 230 value. One point to highlight is that the ease of operation. Spin column technology has been optimized that is easy to use, and in fact it can be just broken down into 5-step process. When you run through the protocol, you'll find that the ease of operation is an advantage. Second solutions that we have, and also our new kit, is called the MacBeats Fast DNA Kit for our feces. So these are the components for our MacBeat kit. As the name suggests, is magnetic beads technology that's involved in the DNA extraction. This will involve the use of the magnetic rack, which we will be able to supply to you. We'll be able to lend you to have a test trial on it, on how this kit can actually fit into the samples that you are dealing with. And right now we are looking at a 6-step process, very similar to Spin Easy, but this time around we are using the concept of magnetic beads. So there will be the use of magnetic rack or even a magnetic rod. So in this case, you go through a pre-wash process, you lyse the cells, the similar concept, and also the optimized lysing matrix that we have. And we purify through the PPS, which is the idea to purify by protein precipitation, centrifugation again. And this is where it differs from Spin Easy and MacBeats. We have the use of magnetic rod or magnetic rack to remove the contaminants and elute the DNA. And on top of that, the biggest advantage for magnetic beads is the automation process. This kit will enable you to operate it in an automatic manner such that you can process large number of samples. So this is a high throughput extraction process. So these are some of the automation instruments that we have tested, and it works well with the kit. So some of the models are the Kingfisher, Starlet. Not restricted to all these. If you happen to have a different automation instrument, we'll be happy to get in touch with you to see how we can incorporate the kit and with your machine so that you still can achieve the high throughput effect that we are seeing. So moving into performance. So again, we want to have it tested through a wide range of samples. And here we have different type of fecal samples, the swine, the mouse, the human. And again, numerical yield obtained. And these are high numbers in terms of extracting from a feces kit. And for purity, again, we are looking at above 1.8 and below 2. So these figures, you can see that they are moving to the range of 2. In fact, the highest point of 2, and also lower end, we have 1.8 and also 1.9. So these are good figures to look at for 260, 280. 260, 230 wise, we have above 1 and also some of it crossing the mark of 2. So which is a good sign that the MagBeads is also able to give high yield and high purity for the fecal samples. So in terms of the numerical values, these are healthy figures. We also want to check it back on the gel. So on the gel itself, we have the genomic DNA bands being intact, no smearing, as you can see on the diagram. Top right again, we will see that the bands are displayed accordingly, and also there's no smearing. And for downstream processing, PCR of the genomic DNA, we see that all the bands were successfully displayed. So we want to compare again with our competitors to see how MP Bio's MagBead kits stand. So in terms of numerical yield, we stand the highest. In this case, we were able to extract the highest number of yield from our swine feces. We tested other animals, just that for display sake, we just pick one of the fecal samples to compare across the different type of kits. So in this case, there are Tenken kit, there are the Kaigen kit, and also the Omega, which is represented by the T, Q, and O. So for purity, we have also compared with them. So in terms of 260, 280, everyone is hitting above the number of 1.8 and also slightly below the value of 2. Just that Kaigen's kit has the tendency to cross the mark of 2. So that's something to take note. For our MagBead kit, we are able to still keep within the range. And moving forward, we are looking at the 260, 230 purity value. So we are achieving the highest purity in terms of 260, 230 value. So across the competitors' kit. So some of the highlights that we have for MagBead's fast DNA kit. So we can use it across a wide range of applications suitable for different feces. Yes, high performance. The quality of DNA is good. Intact bands were displayed, no RNA contamination, adequate yield with consistency. We have high purity, also 260, 230, highest among the competitors. So the ease of operation is the magnetic beads technology, which can be used with automation if possible, and also it offers high throughput. So I have a summary for the whole DNA extraction process. So first, we have the sample collection and also the preparation method, which is using the instruments to lyse the cells and also purifying it in the midstream where you apply. Now when you use the DNA kits. So DNA kits itself, one is the glass milk and one is a spin column, and also it can come in magnetic beads. So they have different targeted applications, different target challenges, and also different targeted samples. So we will have to work accordingly to see what are the different difficulties that end users are facing, so that we can match the exact DNA extraction kit to the individual case. And once you have successfully extracted your DNA, you get it in pure form, then you can send it for 16S sequencing or shotgun sequencing to analyze. These are some of the references that I have for today's webinar. And that's all for my slides. So right now I will invite my colleagues from the sales team and myself to answer any of the questions that you have in mind. And once again, thank you for your attention.
Thank you, Yong-Sin. That's very interesting, and thank you for your time and preparation. We do have a few questions. I think you should be able to see them on the panel. We have a question about a low 260-280 ratio for extracted DNA. Any suggestion on this? So are you able to comment on what might cause this ratio to be low, and any suggestions for overcoming it?
Yes, so that's a good question. So in fact, that's a very common challenge when we are working with feces samples, fecal samples. And one of the suggestions that I would say is to highlight is the protein removal. So we have to ensure that during this process, the contaminants, the protein removal process is done well, to make sure that the step where you have the inhibitor removal, or in fact the protein removal, to have well mixed solution. And also if needed, you can actually keep your samples in the fridge for another 5 minutes. That's coming from that we are using our own kit processing troubleshooting. So we can sort of freeze the sample a bit, cool the sample a bit to make it more precipitated, such that you can precipitate more of these contaminants for some of the tougher samples. And in fact, for one of the troubleshooting we have is also we make sure that the washing step, we want to carry it out twice. We want to ensure that the ethanol is added into the bottle, because these are some of the common mistakes that sometimes people will make when they are doing their DNA extraction. So a couple of troubleshooting there that you can actually pick up that to try and see whether it improves your 260-280 ratio.
Thank you. Okay, so the next question is about low yield and diluted samples. So if samples are diluted and giving low yield, what recommendations do you have to increase the yield?
Yeah, so for diluted samples, the first thing that comes to mind is that you will have to centrifuge a sample at high speed so that you can remove as much supernatant and process the solid precipitate. So by processing the solid precipitate will give you a higher chance that you can be able to achieve most of the DNA that is present in there, rather than processing the whole solution as a whole. So once you have these centrifuge solid particles, you will be able to resuspend them in lysis buffer, and then transfer them into the lysing matrix again, and then you can proceed on with the subsequent extraction steps. And to minimize the loss of sample, in the case where your diluted sample is a fecal sample, we recommend you to skip the pre-wash step and increase the homogenizing speed. So by skipping the pre-wash step, you will also sort of remove the possibility that the DNA can get washed away, or you're removing precious DNA that is already present in low amount for your diluted samples. And also increasing the homogenizing speed, it gives a higher chance and more movement and the force to break the cells, especially when you are already having low yield from your samples. And also one thing, if you're using our kit, we will recommend you to transfer the solution of the binding buffer, the whole solution mixture, to the column. Because sometimes kits will just assign a certain volume to transfer the volume into the column. But in some situation, especially when you are facing low yield, you can try to transfer everything and spin down all the DNA to the column.
Oh, thanks for that. And I mean, I like you know, that your answer involves some modifications of the methodology, and it shows that even though we have a defined protocol, there is flexibility within the protocol, and we have the technical team to support you. So if ever you have a specific issue with your project, you can always contact us, and we'll be able to suggest maybe just a small modification of the protocol that will improve your results. And we have a question: why is yield lower for automation when using the beads? I think that's referring to the mag beads kits, Yongxing, in one of your slides that that was comparing automation results to other results.
Yeah, so I would comment that the use of automation is sort of like a more convenient and an ease of operation for large sample size. In this situation, when we look at yield, sometimes the manual process, we are able to control the process, and also the idea that you're handling them one by one. I think that will actually help to improve the yield and also getting more specific on the extraction process. Of course, we can trust the automations to give us the consistency and processing a large number of samples. So I think this is one of the challenges that people are facing in terms of large number of samples to achieve the consistency and also still, you know, the accuracy. So this is a balance between a large number of samples that you're dealing with and the consistency and the accuracy that you can get.
I hope that answers your question. So there aren't any more questions there at the moment, but we still have a few minutes. So if anybody would like to post a question, please do so now. And of course, if you think of something afterwards, you can always contact us at the email addresses for New Zealand and for Australia. So on that note, while we wait for any late questions, you can see on the slide now the contact details for MP New Zealand, and also we're copying this into the Q&A panel as an announcement for you. So for New Zealand, you can contact Adolf Goldwyn for technical assistance or New Zealand customer service, and MP Australia customer service for both issues. We'll handle the technical inquiries as well here. And of course, for the samples, use any of those contact details, or you can find our sample request form in our website. And you can see that also on the slide at mpbio.com.au, live, etc. So you grab that link, and then you can use that to request samples online. So I see a couple more questions have popped in, Yangxing. So let's have a look at this. So let's see. So somebody has issues with lysing of Kombucha scoby. That's a new one for us. Would you like to give our attendees some advice on lysing kombucha?
Yeah, so for this question, interesting one. Kombucha is a popular product. So for the lysis of kombucha scoby, we can look into the different metrics, lysis metrics that we can recommend you. We have a list of lysis metrics, not just matrix E. The one that I mentioned in the slides, lysis matrix E, is designed especially for soil feces sample. So for the case of kombucha scoby, we can find out more information on what are the targeted kind of maybe bacteria that you're looking for. If you're generally looking for bacterial lysis, we can recommend you specific matrix, lysis metrics to go along with your sample. At the same time, we can help you to troubleshoot in terms of the homogenizing step, see how much you can work around the speed, duration. And I think that these are two components that will help you out in terms of lysis of your kombucha sample.
Okay, I hope that answers the question for you. Okay, so I see no further questions. We're 10 minutes ahead of time, which is never a bad thing. So I think on that note, we will like to thank you all again for your attendance. Remember to contact us for the free samples if you want them. And yeah, we look forward to talking to you about your projects in future.